Hi, I am continuing with the gene bank. Another type of gene bank is called a field gene bank. It is not actually the conservation of something in refrigerator or low moisture condition. It is like conservation in living collections, but it is not termed as the botanical garden. It's different. Botanical garden have the visitors all the time, and they will conserve plant in such a way that it will be more presentable to the visitors, and it has economical importance, and it has tourist value. So unlike that, we have something called a concept called a field gene bank. It is actually collection and planting at another location. It may not be a touristy location, and as I told before, like in the botanical gardens, they will always look for plants which look different, and they have some exotic plants will get more importance. But the field gene bank is not like that. Plants are stored in the living condition. They will be replanted, actually replanted to another location. If there is a habitat loss and everything is going on, we'll take some part of plants from one location and plant it in a different habitat. So that's what is a field gene bank is. One place with some plants, and these plants will be protected. It's a collection and a planting in another location. It is used for the conservation of long-lived perennial plants which cannot be conserved in the seed bank, and recalcitrant species which is also cannot be stored in seed bank, and vegetatively protected propagated species. So all those species that cannot be stored in seed bank is protected by field gene banks. Okay, it is commonly used for rubber, coconut, banana, tuber crops, tropical and temperate fruits. Some of these plants don't make seeds, and some of them have they are perennial plants. They have long life cycle. Coconut and so this needs some other strategy other than seed bank to conserve. And vegetatively propagated crops like onion and garlic can be protected like this. They are not making seeds, so they should be protected in field gene bank. Uh, but the field gene bank, unlike the botanical gardens, don't have that much visitor value, but it has large educational value and research value. It used for the plants which cannot be conserved by seed bank, as I told before. It will require, but unlike the seed bank, it will require large space. So it is actually open field. So we are conserving plants which those are large and long life cycle, or difficulty to maintain and protect from natural disasters. Some plants are like that, or plants which are producing little or no seeds can be conserved like this, and some plants are more susceptible to diseases, such plants can be conserved in field gene bank in a protected way, or plants which require controlled pollination for regeneration also protected in this field gene bank. So it is actually for the protection of the plants, conservation of plants which cannot be done in seed bank. Next type of conservation is in-vitro conservation. It is also a part of gene bank, but in this case we are not conserving seeds. We are actually conserving explants. Explants in a sterile, pathogen-free environment. Like it's like in-vitro cultivation. I believe you all know what is a in-vitro cultivation is. We can cultivate plant cells, tissues, or organs of plants and animals in artificial culture media. So those type of cultures can be conserved, and sometimes we can actually make the whole organism from these cells. So those type of cultivation conservation is called in-vitro conservation. We are not actually conserving the natural plant. We are conserving artificially cultivated varieties or parts. It includes establishment of tissue culture and their storage under controlled conditions. Different materials can be stored in vitro conditions, including protoplasts, cells which are grown in suspension or semi-solid cultures, meristem culture, organized plantlets. Everything can be conserved. Anything which you can culture out in a culture media can be conserved in vitro conservation. And the common principle is that if the genetic stability is high, genetic stability is increased as the complexity of the culture material increased. If you are conserving cells, that is the least complex commodity, it will have the least genetic stability. But if you are conserving the plantlet, which is more complex in nature, so the genetic stability is also high. It has many applications. It can be used for the long-term conservation of recalcitrant, sterile or clonally propagated species. It is an alternative to field gene banks because field gene bank require a lot of space, but this one is not. If the plant or animal material which is costly and limited in quantity, we can actually use the in-vitro storage. If the sample is less stable, which can be stored in this in-vitro culture, the risk of genetic alteration is very less in in-vitro conservation. It ensures consistency in experimentation. If you are doing some experiment, if you want to use consistent samples, we can actually use the in-vitro conserved samples, or it can be used as a reference point or reference material. And in this in-vitro cultivation, cryopreservation is the most commonly used and most effective method of in-vitro storage. So these are the three different strategies which can be used in in-vitro conservation. First one is repeated subculturing, second reduced metabolic rate storage, and finally suspended metabolism. In repeated subculturing, we are culturing repeatedly. Okay, if you are growing the cells in a plate, once the growth is over, we will transfer a portion of this plant or animal, the cells, to the other plate. Then it will grow in another plate. So again, once the growth is over, we transfer it to the another one. So we are repeatedly transferring this to the fresh media for growth in vitro. The thing is that this procedure is very expensive, laborious, it needs a lot of chemicals, lot of manpower is required, and we need to manipulate it continuously so that the chance of contamination is high. As we are regenerating it many times, the genetic stability will be lost. So that is not a repeated subculturing is not a good method of conservation of biodiversity. Next strategy is to reduce the metabolic rate storage. We can use some techniques to reduce the metabolic rate. If the metabolic rate is reduced, the growth rate will be reduced. So the amount of regeneration needed will be less. So in this method offer high genetic stability, reduced labor and expense, less physical intervention. We don't need to subculture again and again. So it can be achieved by low temperature or subjecting it to osmotic stress or addition of growth retardants, some chemicals which will prevent the growth of the species or cells. And finally, we have suspended metabolic state storage, which actually suspend or block the metabolism while in storage, and which offer greatest genetic stability because the whole metabolism is suspended for a while during the time of storage. So there is no growth, no metabolic activities, nothing is happening during the storage. So it is one of the best method, and most commonly used suspended metabolism strategy is storage at liquid nitrogen. It's a cryopreservation method. So these are the methods for in vitro storage. First one is cold storage. As the name implies, we are keeping the culture materials in low temperature, in non-freezing temperature actually. Then when we are storing it in low temperature, aging of the material will be slowed down. So we can actually reduce the repeated subculturing. Then low pressure storage, the tissues or cells will be stored in low pressure. When the pressure is low, the partial pressure of oxygen will be also low. So they won't get enough oxygen for respiration and growth. So the growth rate will be reduced. So these are the methods to reduce the growth rate. Low oxygen storage, the pressure will be normal, but the air around the sample will be filled with inert gases like nitrogen, so that the oxygen pressure will be low, so growth rate will be low. It is useful for both long-term and short-term storage of tissue, cells and organs. Cryopreservation, which is the best method, it is done in ultra low temperature, which is around 196 degrees Celsius, minus 196 degrees Celsius, which is the temperature of liquid nitrogen. When subjected to such a low temperature, the whole chemical reaction will be arrested completely. There is nothing is happening in the cell. And in order to protect the cell at such a low temperature, we will use a class of chemicals called cryoprotectants. One example of cryoprotectant is DMSO, dimethyl sulfoxide. It offers greater security for long time, and it is cost effective when we are comparing it with the other methods. And it has prolonged shelf life. We can store things for thousands of years in a cryopreserved manner in liquid nitrogen. Thank you.